Hello students, am I audible and my screen is visible? Anyone? Okay, thank you, Abhija. So shall we start the session? So we will start the session and other people can join later as of now when they come. So my slides are visible. It's my slides are visible. Anyone can respond by unmuting the mic. Okay. So shall we start the session? So good evening to one and all present here. Myself, Ajit Kumar V, a PhD scholar from IIT Guwahati. So this session will focus on week 2 live interaction session on the course Environmental Biotechnology, and the course code is NOC 25BT83. So this week we will cover the recap of the week 2 lectures that we have seen, and I hope you have all gone through the week 2 session of the Environmental Biotechnology course. And this session will be of two parts. First, we will recap the week 2 lectures and what are all the important topics that have been covered. And the second part will be we will be discussing some MCQs along with their detailed solution. So, before moving on to the course Environmental Biotechnology, so I will introduce myself. Myself Ajit Kumar, a JDP program, which is Joint Degree Program, a collaborative research work between IIT Guwahati and IIT BHU. I work in the laboratory of biochemical and environmental engineering, where our lab focuses on various wastewater treatment from various industries. And especially my area of interest is applying the enzymes, different type of enzymes in wastewater treatment, especially textile wastewater treatment. So moving on to my qualification, I have done BTech Biotechnology from Sastra University, Tamil Nadu, and MTech Industrial Biotechnology from NIT Surathkal, Karnataka. So my work experiences, I have worked in many pharma companies like Biocon Limited, Enzyme Bioscience, where I have worked in different departments in R&D and QMS, etc. So this is all about my introduction, and you people, two or three people can introduce themselves by unmuting the mic, or if you are not comfortable with that, you can put your message in the chat box so that I can know the audience. You can introduce yourself by telling your name, your degree, either BTech, BSc, or MSc, your discipline, what kind of discipline are you, biotechnology or life science background, then what college.
Sir,
Anyone? Yes, yes, Ramsey.
sir, good evening. I am Mr. Vamsi Krishnan, presently working in Erode Sengunda Engineering College, Assistant Professor.
Oh, okay, okay. Okay.
Okay, nice to meet you, sir.
Thank you, Ramsey.
Thank you, sir.
Okay. Anyone? Okay, Darshan. Abhija. Like which background you are, like Abhijat, Chayanika, Darshan, which background, like what kind of discipline you are belong to, like whether biotechnology, bioscience, or biotech, MSc botany, Cotton University. Okay. So nice to meet you guys. We will continue with the session. I hope you have all gone through the week one and the week two lectures. And in the week one, what we have covered is the basics of environmental biotechnology and what is the importance of biotechnology in aspect of environment. And we have gone through all the basic terminologies that prevail in the environmental biotechnology. And also we have seen what is microbial ecology and why it is important to study. And we have seen what are the important components in the microbial ecology, where we have seen two major components. One is biotic and non-biotic factors. And also we have seen microbial association, how they help each other or compete each other likewise. So I hope you have all gone through the week one and week two courses, and we will recap here the week two courses of environmental biotechnology. And before moving on to that, anyone can tell what is the difference between biotic and abiotic component in an ecosystem? What does it really means? Abiotic refers to what, and abiotic refers to what? Anyone? Biotic components and non-biotic components in an ecosystem. You can post your answers in the chat box also. Okay, Chayanika, biotic refers to living organism and non-living components. Yes, exactly. Any examples on biotic and abiotic components? Biotics refers to living organism, so it includes anything, microorganism, bacteria, plants, animals. Every living organism belongs to biotic component. Whereas abiotic components means like it includes pH, temperature, water availability. So these are all non-living components that helps in surviving this biotic components. So these are all the basics that have been covered in the week one lecture of environmental biotechnology. So today we will be seeing the week two lecture, where we will start with the basics of species. So species is nothing but a building block of all the life on earth. Like if you take, for example, in a class, there are 20 to 30 students. So every student has a particular name, right? So this particular name refers to giving a unique identity to individual class members in a classroom. So likewise, in an ecosystem, every organism has a species name that is giving an identity to the particular species. So species is nothing but a fundamental unit of biological classification, and the scientists, they are trying to understand the species in order to describe, understand, and compare biological diversity at different levels. So, what do you mean by species? And this is the fundamental unit of biological classification, and it includes, it can be plants, animals, microorganism. If it is a microorganism, E. coli is a species, one of these species. If we take animal, humans belongs to Homo sapiens. So these are all the examples of the species. And in the left hand side, you could see a beautiful forest area where multiple species at different level, whether it's a plant, animals, aquatic species, some microorganisms will also be present. So this is a example for a different species that prevail in an ecosystem. where different species helps each other and compete with each other in order to survive in this environment. So this is the basics of species. And next, how do you measure this species diversity? First of all, we have to understand the term species diversity. So what do you mean by diversity? Diversity means it has some variety or diverse variety of species in a particular region. So this indicates the species diversity. That is how much variety of species that prevail in a particular area or a region or in a given ecosystem. So for any microorganism to survive, what are all the two things we need? Anyone? In order to survive any species, what you need? Anyone? You need nutrients, appropriate nutrients, and environmental condition. So these are all the two important components that a microorganism survives. So if we see the diversity, there are two types that we can measure this variety in an ecosystem. First is species richness. Richness indicates how many number of species are there in a given ecosystem. That means how many types of species are there in a given ecosystem. Whereas the second term, species abundance, refers to frequency of each species. That is how much of each type is prevalent in a given ecosystem. So the first, richness, refers to how many numbers of species, whereas abundance refers to the frequency. So if we take a species diversity, and how do we measure it? By analyzing two important factors, richness and the abundance. So we can take one example. So in the right hand side, I have two community, community 1 and community 2, where we could see four types of trees that are present in both the communities. So if you want to describe the species richness, in both community 1 and 2, we have same number of species richness. Why? Anyone? Anyone? In the right side diagram, we have community 1 and 2, where I have said the species richness is same. How? What does that richness specify? How many types of species that are present in a community. So why it is same in both community 1 and 2?
Because the same number of species are there in two communities, sir, whereas in community 1 and community 2 also. So that only the richness also same.
Yes. There are four types of trees in both communities. That is why species richness is same. Correct, Vamsi? That is correct.
Thank you,
So,
sir. Thank you.
uh,
Thank you, sir.
okay. So what about species evenness or species abundance? It is different because if we see in the community 1, there is an equal proportion of trees that prevail in the community 1, whereas in the community 2, 70 percentage of one species dominates over the other. So there is a difference in the abundance of species. There is a difference of abundance in the species evenness or the abundance, whereas the species richness is same. So from this two community, we understand that community 1, they are more diverse because the four different species has been distributed evenly and it seems to be balanced ecosystem. So this is the basic difference between the species richness. Richness refers to number of species that are present in an environment or an ecosystem. Whereas abundance relates to how many number of species. For example, here in the community 2, in the community 2, one type of tree has 70 percentage dominance over the other trees. So here there is a difference in the abundance and same species richness is there. So if we combine the species richness and abundance, it helps us to measure this species diversity. So this is how they measure the species diversity. Then, after understanding how it is measured, then we have to know why it is important to study this species diversity. Why it is important? The first important point is that it helps in buffering the environmental stress on an ecosystem. That is, it helps in balancing the ecosystem by avoiding the stress that is created on the ecosystem. That is, it helps in balancing the ecosystem. For example, in a forest, if we take, uh, if one pollinator, like for example, bees, we need bees in order to pollinate the flowers, right? So if bees disappears, what happens? The whole ecosystem will be collapsed. So in order to balance the ecosystem, the other pollinators like butterflies, bats, other insects are also helps in maintaining this ecosystem. So in that case, they might take over the job in order to have a correct pollination and balancing the ecosystem. This is why the species diversity is more important in order to have a balanced ecosystem. And the second point is to provide a variety of diets for an organism in an ecosystem. That is, we need foods to survive. If ecosystem provides many food options, this means we will keep the food chain or food web very strong. So it gives a variety of diet for individuals. For example, if if in a rich, we will consider rich ecosystem where fox is there. So what it can consume? It can consume rodent, fruits, or sometimes it can consume birds also. So for a fox, it has multiple options in an ecosystem. So that the species diversity helps in providing a variety of diets for the organism. And third is aesthetic importance. As we also always know, nature is always beautiful because of this species diversity. And there is a diverse ecosystem can sustain complex ecological interaction between biotic and abiotic component. That is, they support this complex environment and they assist the biotic components to survive with the help of the abiotic component. And we know that biotic components are living organisms, whereas abiotics are non-living components that prevail in the ecosystem. For example, if we take a tree, the tree will release O2, which helps in which helps for humans, right? So this is how a interaction occurs at different levels in an ecosystem. So this is why the species diversity across different ecosystem is very important to study, and this is one of the core concept for understanding this microbial ecology. So these are all the points that needs to be remembered why species diversity is important, and it is very important to understand. So next, what controls this species diversity? So as we have already mentioned, there are two important factors that we have to consider. One is nutrient availability and other one is the environmental condition. So what do we mean by nutrient availability? It is different types of nutrients. These are all like these microorganisms needs these nutrients for their growth and survival. So this is the basics for any microorganism, or if it is like even a plant, humans or animals, we need nutrients, and these nutrients are for their own growth and survival. And if we see the nutrients, There are different types of nutrients that are available. For example, carbon source, nitrogen source, macronutrients, electron acceptors, inorganic electron donors. So these are all the some of the nutrients that are needed for a microorganism or any other species to survive. So the next important factor is that the environmental condition, which includes temperature, water potential, pH, oxygen, light, and osmotic condition. So why environmental condition is important? And it is, we have to know that every microorganism or a species has different environmental condition. And for example, if we take a temperature, there are different types of species that grow in different temperature, and also pH. So anyone can tell the definition of alkali or acidophiles? What does this acidophiles means or alkaliphiles? These are all organisms, right? A group, a type of organism. Acidophiles, alkaliphiles. Anyone? Acidophiles. These are all the microorganisms that can grow in what condition? Acidophiles. In the name itself, yes. Acidophiles, they thrive in highly acidic environment. Correct, Abhija, Tuhin. So, alkaliphiles means it will grow in the alkali condition. So, depending upon the microorganism and the species that we see, we have to maintain different condition. And if we take in respect of a temperature, there is thermophilic microorganism, mesophilic, and saccharophilic, which indicates that the thermophilic microorganism can thrive in higher temperature, more than 60 to 120 degrees Celsius also. Whereas mesophiles, they need a normal temperature like 25 to 35 degrees Celsius. So this is how the environmental condition determines the survival of the species. So these nutrient availability and environmental condition controls the species diversity. So after understanding the species diversity, why it is important to understand and also what controls it and basics of the species diversity where we have seen the richness and the abundance. And this is the species diversity concepts that we have to remember. And then we will be going on to the biogeochemical cycle, where from the term itself we could understand that it merges three scientific disciplines. First is bio, which relates to the biological process, then geological process, and chemical factors. So these three factors involved together and they help in the ecosystem to survive. So what does it means? It involves living microorganism. Any living microorganism, either it can be a bacteria, fungi, algae. So it undergoes series of chemical reaction that occurs inside the living organism in an with the help of the abiotic environment. So this is why it is called biogeochemical cycle. So it is not a straight pathway, it is a cyclic pathway which helps in circulating the chemical elements from the environment to the microorganism and back to the environment. So it ensures nothing is waste, everything is transformed from one state to the another state in a biogeochemical cycle. And if we see the chemical elements, it can be any chemical elements like carbon, oxygen, nitrogen, phosphorus, sulfur, and it also includes the micronutrients like copper and molybdenum. So these are all the basics of biogeochemical cycle, and it merges three important disciplines. As we have seen, it integrates biological, geological, and chemical cycle. And if we see the biogeochemical cycle, one of the important reactions that we undergo, every microorganism will undergo is the oxidation and reduction reaction. So anyone can tell What do you mean by oxidation?
Sir, oxidation means addition of oxygen and removal of hydrogen, sir. Whereas reduction means addition of hydrogen and removal of oxygen.
Correct, Vamsi. It is a correct definition. So anyone can tell in terms of electronic concept. So this is a classical definition of oxidation and reduction. In terms of electrons, what happens to the electrons in terms of oxidation and reduction? Okay, so in removal of electrons seen in oxidation or reduction? Yes, correct. So oxidation where atoms or ions loses the electrons, whereas in the reduction they gain the electrons. So correct, Vamsi and Tuin. We will see oxidation is nothing but adding the electron or removing the hydrogen. In terms of electronic concept, these are reactions where atoms or ions loses one or more electron in order to catalyze the reaction. Whereas in the reduction, we'll be adding the hydrogen or removing the oxygen. Whereas in terms of electrons, we can define them as these atoms or ions, they gain one or more electrons. So, after understanding the basic concept of oxidation and reduction, so microbes always carry out redox reaction. So, redox means it will be both oxidation and redox reaction. Reduction will be taking place simultaneously. So, these are all the fundamental chemical process where the microorganism helps in transferring the electrons from the one molecule to the another molecule so that the microorganism gains energy and drives the biogeo's chemical cycle successfully. So every microorganism carry out redox reaction and they help in transferring these electrons from one molecule to the another molecule. So the next is the basics of this microbial redox reaction, where we have to understand three major definitions or terminology, where the first one is electron donor, second one is the electron acceptor, and third one is the electron carrier. So if you see the electron donor, these are like molecules. The term itself tells us that electrons are donated. If electrons are donated, then the process is called, yes, oxidation. So here what happens is the microbes oxidizes the molecules. For example, the organic molecule which includes sugars and fats. Whereas inorganic compounds like or hydrogen gas, sulfur compounds and ions also. So any example for sugar? Sugars are nothing but what? Anyone? Sugars are nothing but sugars are hydrocarbons or carbohydrates.
Sugars are also a type of biomolecules only, no, sir.
Yes, yes. It is a biomolecule. A simple carbohydrates. Yes, Abhija. It's one of the biomolecules. Can you list some of the other biomolecules?
Sir, carbohydrates, proteins, lipids,
Fats.
and fats, sir.
Yes, yes. Correct, Vamsi. So, one example for this carbohydrates. Yes, glucose. It is a simple sugar. So, these are the molecules that donates the electrons. Whereas in the next electron acceptor, by the definition, by the term itself, we could understand the electrons get accepted by the molecule. So the molecule that microbe reduces. So one of the classical example is the oxygen. In electron transport chain also, this oxygen acts as an electron acceptor, especially in an aerobic respiration process. Whereas in an anaerobic respiration process, nitrates, sulfates, and carbon dioxide will be acting as a electron acceptors. So anyone can tell the difference between aerobic process and the anaerobic process?
Sir, Aerobic respiration means presence of oxygen, whereas anaerobic respiration means absence of oxygen.
Absence of oxygen. Correct, Vamsi. Correct. So where in the aerobic respiration, oxygen will be acting as an electron acceptor, whereas nitrates, sulfates acts as an electron acceptor in case of anaerobic condition. So if we can take a simple example where glucose in the presence of oxygen, it is converting into carbon dioxide and water. Here we could see the both oxidation and reduction is taking place where the glucose is getting oxidized into carbon dioxide by losing the electrons, whereas the oxygen is converting into water by gaining the electron and addition of hydrogen. And this is where we say that it is a reduction reaction, whereas glucose is oxidized by the microorganism. So this is the simple example of redox reaction where both oxidation and reduction reaction take place. So the next third important terminology is electron carrier. This is nothing but between the initial electron donor to the final electron acceptor, there is a chain of molecules will be present, and these small molecules are often referred to as electron carrier. It helps in transferring the electrons from one molecule to the another molecule. So these are the three important terminology that we have to remember while studying the microbial redox reaction. One is electron donor, one is electron acceptor, and other one is the electron carrier. So till this, do you have any doubts? Till this, we have seen what is species and how do we measure this species diversity and why it is important to study and what controls this species diversity. Then we have seen biogeochemical cycle where reduction reactions are more prevalent, and we have seen some of the basics of microbial redox reaction terminologies. So till here, do you have any doubts? So okay, so we will move on to the next part where if you have gone through the week 2 lecture, sir could have said that microbial ecology and environmental biotechnology, they are tied to each other. And why it is so? Why they are tied to each other? Because these two concepts, they are always inherent. They help each other in order to understand the process. If you see the environmental biotechnologist, they apply the concept and tools of microbial ecology to manage this environmental biotechnology process. So this is why it is very important to study the microbial ecology concept, and it helps in understanding the microbial community and how do they interact with each other. These are all the main important concepts behind the microbial ecology. After understanding these two goals or the concepts of how microbial community exists and how they help each other and also how they interact with the environment is all important. And if we able to understand this concept, we can apply in the real time problems like environmental issues, pollution prevention control, so that we can find a valuable solution. So this is why microbial ecology is very important concept, and it is always tied to each other with the environmental biotechnology. So if we see how it is integrated with the different disciplines, we will see. First, if we see in order to study the in situ gene diversity expression, or you have to identify or find the interaction among the organisms that are responsible for this geochemical process or change, there are two ways of approach you can deal with. One is we can go for lab incubated studies. Another one is culture independent studies. What do you mean lab incubated studies? Like you will be incubating them in an appropriate condition in the lab scale level. What can be done? You can isolate the pure culture in the laboratory. By isolating a pure culture, you could study their growth, how much energy that they can yield from a simple sugars, and what type of enzymes they are secreting, and what kind of mechanism they play in helping these environment, helping this microorganism to survive for their growth and metabolism. And we could also study their physiology, what is their structural components, and how do they help in order for their own survival. And we could study the chemical reactions too by analyzing their mutation, that is changes in their structure in the gene level, and recombination and gene regulation. And then comes into the action of genetics and molecular biology, where you can sequence the gene, which helps in identifying their phylogenetic relationship, that is, how much they deviate or close to one another in a group of species. So these are all like lab incubated, or you need a laboratory in order to perform this experiment. Whereas we could also correlate with cultivation independent. You don't need to cultivate the microorganism. You can directly study the microbial ecology by analyzing the cells in a microscope level. Also, you can analyze the enzymatic activity or cell component. Am I audible?
Yes, sir. You are audible, sir.
Okay, okay. My screen is visible, right? Slides are visible, right?
Slides are now visible, sir.
Okay. So we have been seeing the cultivation independent technique where several nucleic acid extraction can be done, and it can be deducted using various techniques like microarray, metagenomics, fish technique, which is fluorescent in situ hybridization technique, and PCR and RT-PCR. So these are all the different disciplines that needs to work together in order to study this microbial ecology. So we need an biochemistry, molecular biology, genetics, and recombinant DNA technologies in order to these different disciplines are needed in order to study a microbial ecology in a community. So what would be the ultimate goal of studying this microbial ecology? The first will be we have to understand the microbial community. If you take any environment, if it is an contaminated environment or if it is an oil spill environment, whatever the environment, first we have to understand what are all the microorganisms that are present in a given ecosystem, and then how this microorganism not only interact with each other, also with the environment. So if we are able to understand and answer these two questions, then we could understand the whole picture of this microbial ecology. So anyone can tell what is mean by microbial community? Microbial community refers to microbial community.
Sir, microbial community refers to the group of microorganisms live together.
Yes, yes. It's correct. So community refers to two or different species. They prevail and live together in order to form a community. Yes, it's correct, Vamsi. So as we have seen the what is the goal of the microbial ecology, then in order to understand this microbial ecology, we have to answer these four questions. If you are able to answer this four question, then you could understand the microbial ecology very well. So the first question is what kind of microorganisms they are present in the community. And the next question is what are all the capability of the microorganism for carrying out the reaction that transforms the community environment. So this means that what the microorganism can capable of do, what they can do. It answers the question. So the third one is what reactions and transformation are the community members actually perform. Here we are trying to answer what kind of reactions and transformation the microorganism is undergoing. And in the fourth question, what are all the interrelationship among the community member between them and their environment. So this answers the question of how do they interact with each other and also with the environment, which is abiotic components. So if you are able to answer these four questions, then I hope you have understand the concept of microbial ecology. So first we will see the first two questions where what microorganism are present in the community. So what does it means? What does it mean? What are all the it tells us what are all the microorganisms that are present in a given ecosystem. So we will take a simple example of an yogurt or curd. So here we could see that in order to convert a milk to the curd, it cannot be done by a single microorganism. It needs a group of microorganisms, mostly of lactobacillus or staphylococcus microorganism, which helps in fermenting the milk into curd. So here the first question answer the what kind of microorganism that are present in helping converting this yogurt to milk to yogurt. So we have identified most of the lactobacillus species and staphylococcus. Lactobacillus and staphylococcus are responsible for converting this milk into curd. So this is how the first question is answered by telling how many different types of microorganisms are there and how much abundance are there. So the next question is what are the capability of the microorganism for carrying out the reaction that transforms the community environment. Here it answers what can they do. What can they do? We will take this. We will take the same example. We will take the same example like what microorganism, like what lactobacillus or staphylococcus do. In order to answer the second question, these microorganisms in a community, they help in fermenting this lactose into lactic acid. So their job is, it is a dairy fermentation. They have a skill of dairy fermentation. They will be converting the lactose into lactic acid. So this is how the first two questions in answer. First is what are all the microorganisms that are present in a community, and second is what are all of the what these microorganisms can do? They can help in fermenting this milk into yogurt by converting into lactic acid. So the third question is what kind of reactions or transformation actually they can perform. So this answers the question of what actually they are doing in a community. So we will take the same example where we could say that these microorganisms produce, for example, the staphylococcus will produce lactic acid, and this lactic acid is an acid, right? So it lowers the pH. which favors the environment for lactobacillus species, which helps in converting this milk into curd by coagulating them. So this is the reactions and transformation that each individual undergoes, which helps in converting the final product, yogurt or curd. And the fourth question is, what are all the interrelationship among the members as well as with the environment? So here they always have a synergetic action. That is, they help each other. How it helps? This staphylococcus will convert the lactose into lactic acid, which helps in lowering the pH. And this in turn helps the other lactobacillus species to convert into yogurt or curd. So you need an appropriate acidic environment, and also you need help from the two microorganisms. So this is how these four questions is answered. And these are all the important four questions. One is, what are all the microorganisms that are present and what they are capable of do in a particular environment? And third question, what kind of reactions or transformations that takes place in the community members in order to perform this reaction? And the fourth one is how these members helps each other, and also how it gets help from the environment in order to do these processes. So if you are able to answer this four question to any environment, then I hope you have understood the microbial ecology concept very well. So we will take one real-time example and we will try to answer these questions one by one. So this is the textile wastewater, and now we are going to remediate this textile wastewater by understanding the microbial ecology concept, and we will apply it and we will answer these four questions. So we will answer, it will be an interactive session, so you can also answer. So first question is, what kind of microorganisms are present? If you take if you take an environment, it which is contaminated with the textile wastewater, first question is we have to find what are all the microorganisms that are present. So there will be a millions of microorganisms will be present, right from Bacillus species, Staphylococcus species, any species. There will be a millions of microorganisms that will be present in an ecosystem. So how do we find this microorganism? Anyone? How do we find the microorganism in the textile wastewater or any wastewater? In order to study or to find the presence of microorganism in a contaminated area, how do we find this microorganism? Any technique? MPN fish, yes. So the any basic technique? So these are all like advanced techniques. So how do we isolate a microorganism? Like microorganism used to view the cells. So anyone heard of isolating a microorganism using serial dilution method? Yes. So the basic technique is we will be collecting the sample and we will be performing the serial dilution so that we could understand what are all the different types of microorganisms that are present in this ecosystem. And then once we have found out what are all the microorganisms that are present in an textile wastewater, the next question is what they can do. What are their capability? In order to answer this question, these microorganisms can synthesize specialized enzymes which may help in breaking down this complex dyes or salts, chlorinated aromatics into non-simpler products. So these specialized enzymes or proteins that they secrete is the answer of what they can do. And third question is what kind of reactions or transformation they perform in order to convert this wastewater into clean water. So if you are able to understand what kind of reactions these microorganisms undergo or what kind of transformation of this toxic dye into non-toxic dye, so if you are able to understand and answer these questions, then it will be easier to understand the microbial ecology. So what happens here, the enzymes or proteins that they secrete will help in transforming the toxic dye into non-toxic form. And the fourth question is how do they interact with each other and their environment? So here what happens, millions of microorganisms will be prevalent in the environment and they help each other in order to synthesize different enzymes. And these different enzymes, they are synthesis for their own growth and survival. So these are all the teamwork. They share their metabolites and help each other in order to convert and polluted environment into non-polluted environment. So this is one of the basic example where we could apply the microbial ecology concept and we could answer these four questions effectively. So if you are given any environment, it can be an pharmaceutical wastewater or food industry wastewater or tannery industry, every industry or any ecological community or a forest, any oil contaminated site, if you are able to answer these four questions and if you are able to adapt different techniques at each stage, then it's better and it is very efficient in order to face the environmental challenges issue. So these are all the main concepts that prevail in the microbial ecology. So till this, do you have any doubts? So understanding this microbial ecology concepts and these four questions, do you have any doubts? Okay, Abhija. Any other doubt? So okay, we will move on to the next concept. As we have seen, the first question, which is how many microorganisms that are present in the community. In order to find that, this is the answer. We will be isolating the microorganism from the environment. So, anyone, what do you mean by isolation? Isolation or isolation of microorganism means anyone? Have you heard the term isolation of microorganism? Yes, yes, Abhija. It is a separating a specific or a pure microbe from a community of microbe in an environment. Correct, Abhija. So it is a process of separating a single species of microorganism from its natural habitat. So it is a how do we achieve this isolation? Through serial dilution. It is one of the basic technique where we will be diluting the solution to an appropriate level by using a correct dilution factor. And this serial dilution is performed to reduce the concentration of the cells in order to simplify the operation. So if we take the example, so my aim was or my aim is to isolate a microorganism from a environmental sample, soil sample. So what I will do first, I will dig or make a depth of 20 centimeter and I will try to collect the samples because the surface soil samples will contain all contaminants, plastics and all other debris. So we will collect the samples at the depth of at least 20 centimeter and we will take into the laboratory by storing them in a sterile bags. And after storing them in the sterile bags, we will dry it at the room temperature, at a room condition at 35, 30 degrees Celsius. Then if we want some pre-treatment like a thermal microwave sonication, we can perform, otherwise it is an optional. So we will directly go for serial dilution. Where serial dilution means we will dilute the concentrated sample to the diluted. Why? In order to reduce the population and in order to isolate a particular specific microorganism. So we could observe the color change as dilution occurs. And how these serial dilution are performed. So what happens here in the first test tube, we will fill 1 gram of soil with 9 ml of distilled water. Then we will mix it thoroughly. Then we will take 1 ml of the sample and we will be 1 ml of this per sample and we will be adding to the next tube which contains 9 ml of water. So this dilution we refer to as 10 to the power of minus 1. That is, we have diluted 10 times with the already diluted one. So as the dilution proceeds, the concentration of the cells get decreased. And normally they will go up to 10 to the power of minus 6 to 8 to get a single pure colonies. And it also depends upon the samples that we consider. So after diluting the sample, we will take the last one and we will plate it in an appropriate nutrient agar or any specified agar. And here there is an option of adding some antibiotics if you are screening for a particular microorganism. So after adding the inhibitors, what is the role of these inhibitors? They will inhibit all other microorganisms. They will allow only the specific microorganism to grow and promote their growth. So this is how the microorganisms are isolated and we will be incubating them at an appropriate condition. And we could see this is one of the example where quadrant streaking is done and we could see a single pure colonies has been achieved. And further we can cultivate them in a larger scale and we can go for it. So this is the primary and basic example of why and how isolation is performed. Isolation is performed where we are applying a specific condition like inhibitors in order to screen a specific microorganism so that we can avoid the undesired microorganism. So this is the normal and basic procedure of how isolation is done. Then after isolation, the more advanced topics are metagenomics. Metagenomics, so we have seen the isolation part and how it is done using the simple laboratory technique. So if we want to understand the microbial interaction in a different environment, and we know that most of the microbes, they cannot be cultured using this standard laboratory technique because each microbes have different conditions and still lot more need to be discovered in the microbes. And till now, there are new and novel culture techniques has been evolving and it is still in the progress part. So every microorganism we cannot culture. Then what is the way? The only way is we can go for direct metagenomics in the environmental sample or any other sample. So this is a culture independent technique and it is used to study vast majority of the microbes that are present in a given environment. So what do you mean by metagenomics? So it is a study all kind of genomes in an organism. What do you mean by genome? Anyone? Genome means what do you mean by the term genome? Genome refers to
A complete study of genetic chromosomal aberrations is called
yes,
genomic.
genome. So it is a collection of genes. Yes, correct. Ruin, you are correct. Complete set of chromosomes. So if you are able to study all the genes in an microorganism, and the technique that we use is the metagenomics. So it is a study of all the genomes of an organism that has been isolated from the bulk sample. So why it is important? It helps in analyzing the community of microorganism in a different environment. It can be soil, water sample of any samples you can analyze and we can try to understand the community of microorganism. So what does it use? It uses several genomic technologies and tools in order to understand their genomes in a microbial community and how complex they are and how they interact with each other and what kind of genes they are sharing one among each other. So these are all some of the studies they have been carrying out to understand this metagenomics approach. And if you see the process, as we have seen, this is helps in studying the whole genome directly without culturing the individual species. So what kind of process they undergo? First, we will collect the environmental samples, then we will extract their DNA and we will sequence them. DNA sequencing is a vast topic and we will be seeing in the upcoming weeks and we will be sequencing the DNA samples and we will be analyzing this sequence data with help of the bioinformatics tools. So this is the general protocol or the procedure that follows for every metagenomics approach. And here we could see that there are two kinds of approach we can go for. One is targeted metagenomics, one is the shotgun or whole genome. And the first one, which is targeted metagenomics, by the term itself, we could understand it is a targeted. So here we will be sequencing only a specific genetic marker or conserved region within a microbial community. For example, in order to identify a bacterial sample, we will be synthesizing the 16S rRNA because it is a conserved region and this sequence they do not evolve much as across the generation. That is why we consider this 16S ribosomal RNA in order to sequence them and identify any bacterial species. Because in every bacteria has a signature of 16S rRNA and these are different for each species. And if you are able to analyze this 16 srRNA and compare it with the bioinformatics tools, then it is very easier to identify this bacteria. So this is the case of bacteria. So if we concentrate on fungal species, instead of 16S, as these are all like eukaryotic species, these fungus are eukaryotic, right? So if it is an eukaryotic, we will have an 18S ribosomal RNA, or we could sequence this ITS regions, which is internal transcribed spaces for fungal species. So we have to remember if you are aiming for a bacterial identification, you have to go for 16S rRNA sequencing, or if you are going for a fungal species, you could sequence 18S rRNA or ITS regions. So here what happens? It is a targeted metagenomics. We are targeting a specific genetic markers like 16S rRNA and 18S rRNA in case of bacteria and fungus respectively. So if you want to sequence whole genome, and it is not targeted, right? So it involves random sequencing of all genetic material that are present in a sample. So these are all the two important types that prevalent in the metagenomics. And if you see the steps involved, so first step is you will be collecting the sample and you will be processing it. And what happens first, you will be collecting the sample from the environment. As we have seen how to collect the sample, we have to collect the sample at a depth of 20 centimeter. In case of soil sample, we have to put in a sterile bags and transport to the laboratory. And the next one and next processing is you need to extract the DNA using a authorized protocols. One of the protocol is phenol chloroform assay, or a protocol to extract the DNA from the soil sample or water sample or human sample. So then after extracting the DNA, you have to assess their purity. Then after assessing their purity, you can go for the next step, which is the second step. You will be constructing a metagenomics library. What do you mean by constructing? So what happens here is you are having a DNA, so you will be putting these every individual DNA in an vector or an host in order to clone them. First you will be putting in a vector, then you will put in an appropriate host like E. coli in order to go for next step. So anyone, what do you mean by vector? Have you heard the term vector and why it is used in genetic engineering? Any idea? Vector, as we have seen, these genes are integrated into the vector and it has been transformed in the host, right? So what do you mean by vector? Yes, it acts as a it is a tool, it is a delivery vehicle that carries the foreign DNA or your species of interest DNA species. So it is correct, Abhija. It acts as a vehicle or yes, it helps in carry the gene of interest that we are focusing on. So once we have put in an appropriate host, then we will screen this using the metagenomic libraries. How do we screen this? So first we will identify what are all these species specific genes by identifying them using the microarray technique that we will seeing in the upcoming slides, or we can use FISH, which is fluorescent in situ hybridization technique. So by using different tools and techniques, we will be screening these metagenomic libraries. And the next step is we will be sequencing the DNA using next gen sequencing or Sanger sequencing. So once we have sequenced the DNA, we will be going to the next step, which is sequence assembly. Once we have sequenced it, we will be trying to assemble because these are all the bits of DNA which are waiting to assemble. So we will assemble this DNA and reconstruct them. So what happens? We will have a short sequence and we assemble to get an longer sequence. The need of longer sequence is that they provide more information and better accuracy. So these are all the assembled genome sequence. And the next step is binning, where it will sort or assemble all the bits of sequence DNA into a group that represent an individual genome. So this is the step where we will assemble the genome sequence and we will annotate them, and here the genes will be identified and will predict their functions. And the next step will be experimental design and statistical analysis, where we will be using some bioinformatics tools in order to understand their functional genes, their enzymes, their proteins, and we will try to predict their functions behind it. And we will be storing the data and will be sharing through worldwide. So any database that you know, have you come across any database that stores these information, genes information? What kind of libraries are there in order to store these information? Yes, DDBG. Any other? What kind of database? Any other database? Swiss database. So we have NCBI, DNA database like GenBank is correct. We have NCBI, EMBL. So these are all some of the database that you can access at free of cost, where millions of sequences are deposited, and it helps to understand the microbial ecology well. So this is the metagenomic part, where the primary concept is we will be analyzing all the genome that are present in an organism. And how it is done? There are multiple steps involved where we will be constructing the library. After that, we will be screening it. We will be analyzing the DNA sequence. We will try to assemble all the short sequence into a larger assembled genome sequence, and we will use multiple bioinformatic tools in order to analyze their statistical analysis, and we will store these data in a libraries. And by this we could understand what are the genes that are involved, what kind of enzymes they secrete, and how it helps in predicting their functions. So these are all the important concepts in metagenomics. And if you see some of the examples, well-known examples or metagenomic projects, the first one is Human Microbiome Project, where it is launched by National Institute of Health in 2007, and the main aim of this project is to study the microbial community that are present, especially in the human body. So what they will take a mouth sample, skin sample, and gut sample, and they will analyze what are all the bacteria that are present in the gut. And this gut microbiota is a separate section where they will have lot of microbial species that are prevalent in the gut microbiota. This is studied in order to keep our digestive system clean. So this is why it is very important to study the microbial community, and it helps in analyzing the community very well and how do they interact with our body, human body, and how do they contribute and how much we contribute. So for example, this microbiota helps in keeping our digestive system safe, and as well as what we do, we give shelter to this microbiota. This is how uh, we and the microbiota helps each other by giving shelter for both of them. So, moving on to this human microbiome project, as it was launched in 2007, and they helped to study the microbiome community. And this project aimed to understand the role of microbiome in human health and disease. And they sequence and analyze the DNA of microbial community directly from the human samples. So this helps us to understand the microbial community that exists in the skin region, gut, and mouth. So this is one of the important project, and this is achieved through the advent of this metagenomics. And the other classical example is that the global ocean sampling expedition. It is one of the project that was led by Craig Venter and his team in 2004. And this project aimed to understand the microbial diversity, especially in the ocean region, in the marine region, where they used shotgun metagenomic sequencing approach in order to sequence whole microbial community that prevalent in the ocean system. So this project helps us to understand more of marine ecosystem, and it revealed many of the unknown microorganisms and the genes, and this project helps in understanding more of different different microorganisms that are prevalent in the ocean. So these are all the some of the important projects that has been utilized this metagenomic approach and helps in simplifying our job by identifying unknown species and organisms that are prevalent either in the ocean or in the human body. So this is the core concepts behind the metagenomics, and the next concept that we will move through is the microarray. And what do you mean by microarray? This you can imagine like a chip that we see in the semiconductor devices like even if it is a computer or phone, we have chip, right? So it is like a chip like structure where with the solid support which is mainly made up of glass or silicon, upon which we will be attaching the DNA in an organized manner. And what is the main principle behind it? So what happens is this: we will be hybridizing the nucleic acid. Nucleic acid. What do you mean by nucleic acid? Anyone? Nucleic acids are example of nucleic acid. DNA and RNA. And these DNAs and RNAs are made up of four components, right? DNA is made up of, and what is the difference in RNA? Yes, they are made up of nucleotides. What makes DNA? What are the four components that are present? DNA consists of A, adenine, A, T, C, G, guanine, cytosine, right? Then what is the difference in RNA? So DNA is made up of A, T, C, G, whereas RNA is made up of, there is one difference, right? Yes, so again it is made up of pentose sugar, nitrogenous phosphoric acid. Yes, the difference is uracil. So this is the basics of nucleic acid, and what happens in the microarray. These nucleic acid, due to the complementary base pairing approach, they bind to each other. They use the property of complementary nucleic acid. So, so what binds? So A will always bind with, in a DNA, A will always bind with, yes, T, and C always bind with G. Yes, correct. So this is the nucleic acid base pairing approach, complementary nucleic acid sequence. They specifically bind to each other through hydrogen bonding. So this is the basic principle of the microarray. What happens? There is a hybridization between the nucleic acid strands. So if we see the nucleic acid technique, microarray technique, the main important criteria is you have to label the samples using the fluorescent dyes. The fluorescent dyes can be green fluorescence, red fluorescence, green fluorescence. There are different types of dyes prevalent that stains the samples. So you need to label the sample, and you need at least two samples to hybridize into the chip. And we have seen that the main principle is that complementary base pairing between the sample and the probe. In probe, In probe, you will have nucleic acid where you will be applying the DNA sequence on it. So if there is an base pairing, complementary base pair that occurs, they will bind to each other. And any other non-specific bonding, they will be unattached and they will be washed out during the washing step. So what happens here? The fluorescent labeled target sequence that is our sequence which is already labeled, they will bind to the probe sequence that is present in this chip and it will generate a signal. So depending upon the signal, we will get to know what are all the amount of target sample present. If suppose the signal is very huge and it is large, then a large amount of sample is present in a given system. So we will take one example. We will see one is healthy cells, one is pathological cells. So we will take a cell and we will culture it and we will try to isolate the RNA. After isolating the RNA, we will fluorescently tag this RNAs. Before that, we will reverse transcribe it in order to convert this DNA into RNA into DNA. Then this DNA will be fluorescently tagged. For example, if it is a healthy cell, we will be tagging them with the green dye, fluorescent dyes. If it is a pathological cells, we will label them with the red dye. So we will be attaching them on the microarray sample. So once we apply the sample, if we get a green signal, then it indicates that it is a normal cell and this cell is free of contaminants or it is not diseased cells. Whereas if our sample is in red color, then it is a pathological cell. It tells us it can cause disease. So if it is an yellow color, so it is a presence of both cell. There is an equal ratio of both presence of healthy and pathological cell. So this is how we will find whether the cell is healthy or it is a cancer cell. Healthy cell or normal cell. So this is one of the main technique or important technique that you have to be understood for microbial ecology. So, as we have seen the concept and techniques, how do we use this microarray technique in the ecology? So, what happens is by analyzing the sample, by analyzing the samples, so here what they have done is they have took one oil contaminated sample and they have conducted the microarray. This is geochip hybridization and they have found out what are all the abundance of the bacteria species. So here we could see acetobacteria, alpha proteobacteria, beta proteobacteria, gamma proteobacteria, delta proteobacteria. So by applying this microarray technique, we have answered the first question, which is what are all the microorganisms that are present in a given ecosystem. And here the ecosystem refers to oil contaminated sites. And from doing so, we could identify these are all the bacterial samples. And the second question is how much abundant they are. We could measure the intensity in each sample. So this also answers the species diversity. One is species richness and species abundance. How much, how many type of species are there and how much prevalent in that particular microorganism. So this is the first image predicts the only the bacterial species and the second tells us the archaeal abundance. We can see that DQ2 which is one of the site where the more 20 the signal is huge. There is large abundance of archaeal species prevalent in the DQ2 and same the fungal species. So here we could see that in all three species, the DQ2 region has diverse species community. If you see, more abundant of bacterial, archaeal, and fungal species. This is how you have to apply the microarray in order to study the ecology. And second image is telling us that what kind of genes that are responsible and also for carrying out this reaction. So as we have seen a biogeochemical cycle, right? So here we have seen carbon cycle, examples of a biogeocycles are carbon cycle, nitrogen cycle, etc. Right? So in order to assess the carbon cycle, these are all the some of the enzymes that are responsible for carrying out this carbon cycle. And these are all the enzymes that are responsible for carrying out this nitrogen cycle. And in order to decontaminate or degrade this oil spills, these are all some of the organisms that help in, these are all the enzymes that help in driving this microbial decontaminating site into a non-polluting site. So this is how microarray technique is applied in the ecology, applied in the technology, and this is how you understand the microbial community by analyzing their abundance and richness and how do they interact with each other and how they prevalent with in their given ecosystem. So till now we have seen how do we isolate the microorganism and what is the importance of metagenomics and how this metagenomics is done and what are all the examples projects that metagenomics has been used. And third topic we have seen is how microarray technique is done and what it is and what is a principle behind it. And we have seen how it is applied in the microbial ecology concept as well. So this is the concept, and these are all the important concepts that has been gone through in the week 2 of the course environmental biotechnology. And I hope you have all understood the concepts, and if you have any doubts you can ask me, and if there is no doubt we can proceed with the MCQs, sample MCQs. Is there any doubt? And these are all the important concepts in the week 2 lecture, and if you haven't gone through you can go through once and understand even more better. And in week 2 we have seen many important concepts and we have answered 4 important questions, and we understand the microbial species diversity and how it is important to understand the richness and abundance and how do we isolate the microorganism and what are all the different integrated disciplines that drives the microbial ecology concept. Then we have seen metagenomics and we have seen the microarray techniques. So these are all the some of the important concepts in the week 2 lecture. Do you people have any doubts? Any doubts? I hope you don't have any doubts. And if you have any doubts, You can email me or you can post your questions in the discussion forum so that we will address those questions also. You can go through the week 1 and week 2 lectures, and if you have doubts in those lectures, you can mail us or you can discuss in the discussion forum. So I hope we can move on to the MCQs. So it will be an interactive session. So I will read out the question. You can post your answers in the chat box or you can unmute the mic and you can answer those questions. So we will move on to the question. The first question is, what are all the super challenges of 21st centuries? The option A, climate change and energy supply. Option B is health disease. Option C, sustainable environment. Option D, all of these. And I hope this was discussed in the lecture 1 of the week 1 course. So what are all the super challenges of the 21st century? What is the answer? Yes, it is correct. The answer is D. All of this has been discussed in the lecture 1, week 1 of the environmental biotechnology, where climate change, energy supply, health and disease, sustainable environment, all the super challenges that needs to be tackled, and how it is tackled using environmental biotechnology tools, we will be seeing in the upcoming sessions. So as of now, all of these are the answer, and we will move on to the next question. So which of these following comparison of eukaryotic and prokaryotic cell is false? I will repeat it. Which of the following comparison of eukaryotic and prokaryotic cells is false? Option A, eukaryotic cells are larger than prokaryotic cells, though some exceptions apply. Option B, prokaryotic cells have unorganized nuclear materials compared to eukaryotic cells. Option C, mitochondria and chloroplast reassemble prokaryotic cells. Option D, ribosome structures is different in eukaryotic and prokaryotic. So you please go through the question. Which of the following comparison of eukaryotic and prokaryotic cell is false? Which of the statement is false? You can post your answer in the chat box or you can unmute the mic. So any answer? Which of the following comparison? Option D. Yes, option D, which is prokaryotic cells have membrane bound organelles while eukaryotes do not have. We will see one by one. If in the case of option A, eukaryotic cells are larger than prokaryotic cells. Yes, it is correct because eukaryotic cells has a diameter of 10 to 100 micrometer diameter, whereas prokaryotics are very small. They have only zero point 0.1 to 5 microns in diameter. So the first statement is correct, where eukaryotics are larger, that is 10 to 100 micrometer, and prokaryotic cells have unorganized nuclear materials compared to eukaryotic cells. Yes, it has. The prokaryotics, what do they have? They have a DNA that is located in the nuclei. They are present in the cytoplasm itself, and this DNA is more concentrated. So they do not have a well developed nuclear membranes as we seen in the eukaryotic cells. And in case of option C, mitochondrial chloroplasts resemble prokaryotic cells. How does? Because both of these, they are same in the size, structure, and also these both mitochondria and chloroplasts, they have own ribosomes. and these ribosomes are also similar. That is why mitochondria and chloroplast resembles prokaryotic cells. And as we have seen, prokaryotic, they do not have a membrane-bound organelles, and their ribosome structures are different. For example, in prokaryotic, their ribosomes are made up of 80S, and they are larger and complex. Whereas in prokaryotic, they are made up of 70S structure, and they are simple and small. So if you see all these options, yes, the prokaryotics, they do not have a well membrane bound organelles. Whereas if we see the eukaryotic, they have nucleus, well developed nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, lysosome, and vacuoles. So as we seen in the picture also, they have well developed membrane bound organelles, whereas prokaryote they do not have. So the option is D. So we will move on to the next question. The next question is, which of the following statement about biotechnology product is false? Option A, human insulin is synthesized using E. coli cells. Option B, Bacillus anthracis toxin is used to protect the crops from the grazing animal. Option C, Clostridium botulinum toxin is used for cosmetic and medical purpose. Option D, Pseudomonas petuda is genetically engineered species that are capable of cleaning the oil spills. And option E, genetically modified adenovirus is used to deliver the vaccine against the various disease. So, which of these following statement about the biotechnological product is false? Answer. Hello? Am I audible? My screen is visible?
Yes, properly you are audible and visible too.
Okay, okay. Thanks, Prince. So, as we have seen, I will repeat it once. Which of the following statement about biotechnological product is false? Answer. Option B. Any other answer? Option B. Any other? Yes, the answer is B, which is false. Bacillus anthracis toxin is used to protect the crops from the grazing animal. And here we will see Bacillus anthracis, it is a gram-positive bacteria. It is a causative agent for anthrax. And this organism is often toxicogenic, that is, they cause toxic. And it secretes two toxins. And these two toxins are made up of three proteins. And these three proteins includes one protective antigen, one lethal factor, and one edema factor. So this Bacillus anthracis, they secrete the toxins, and particularly it is very deadly to the humans and the animals. So that is why it is not used for agricultural purposes. That is why Bacillus anthracis toxins are used to protect crops from grazing animals. So we will see other options too. Why it is correct? Human insulin is synthesized using modified E. coli cells. Yes, it is human insulins. What do you mean by insulin? Insulin is used to regulate the blood sugar level. It helps in treating the diabetes, right? So, in order to synthesize in a larger quantity, they use E. coli cells through recombinant DNA technology. And that is how human insulin is produced in a larger scale using the E. coli cells. And option B, as we have already seen, it cannot be used to protect the crops from grazing animals. And option 3, which is clostridium toxin, yes, it is used for cosmetics and medical purpose. Where in cosmetics, these toxins are used to for wrinkle wrinkage and facial asymmetry corrections. Whereas in the medical purpose, they have been used in treating the chronic migraines, cerebral palsy, and sclerosis. So this is how this Clostridium botulinum toxins are used in different industries like cosmetics and medical purpose. And Pseudomonas pituita, which is often referred as superbug, which is a genetically engineered species. This has been used for cleaning the oil spills. And oil spills are nothing but these are all hydrocarbons, liquid hydrocarbons, and these Pseudomonas pituita helps in cleaning these hydrocarbons, converting them into non-toxic form. And the third option, genetically modified adenovirus. This acts as a vehicle, which is vector, in order to deliver the vaccine against the various disease. So among this, option B is false, and you all said correct. The option B is correct. So we will move on to the next question, which is a thermophilic bacteria from hot geyser of Yellowstone National Park, USA, was the source of valuable enzyme. So, which is a thermophilic bacteria that has been isolated from hot geyser of Yellowstone National Park? Option A, pepsin. Option B, tyrosine. Chymosin. Option C, D, 529 DNA polymerase. Option E, tag DNA polymerase. So, what is the answer? Which is the thermophilic bacteria that has been isolated from hot geysers of Yellowstone National Park? What is the correct answer? It is pepsin or tyrosine or chymosin or 529 DNA polymerase or tag DNA polymerase. Option E. So, what is the answer? Option E, tag DNA polymerase. So, Friends, what type of bacteria prevent from grazing animals? So, in order to prevent from grazing animals, we can use Heterobacteria and other species. And what's the eddy factor? That is not eddy factor, the edema factor. So these are all the components. If you see the toxins, this is one of the protein in the anthros, in the what? Bacillus anthracis toxins. So it is one of the protein that assist in making them toxin. So this is edema factor, and Heterobacteriaceae and a lot of bacteria is there. I hope I have cleared your doubt. So we will move on to the next question. And as you have said, the answer is correct. The tag DNA polymerase. And anyone, what do you mean by thermophilic bacteria? What are thermophilic bacteria?
A bacteria resistance to temperature.
Yes, it can survive in the higher temperature. So from Thermophilus aquaticus, this is a bacteria where they have identified this tag DNA polymerase. And what is the role of DNA polymerase? Anyone know? What do you mean by DNA polymerase? What is the role of DNA polymerase in any reaction? DNA polymerase, what does it do? The DNA polymerase role is to add nucleotide to the strands. Okay, then if we see the first three options, what are they? Pepsin, tyrosine, chymosin. These are all what kind of enzyme? These are all proteolytic enzymes, which helps in degrading larger protein into simpler amino acid. And this 529 DNA polymerase, it is also one of the DNA polymerase that has been isolated from Bacillus subtilis. So among this, tag DNA polymerase is the thermophilic bacteria. So as we see, thermophiles are extremophiles. They can survive in higher temperature between 41 to 121 degrees Celsius. This is separated from the Thermus aquaticus, and it was discovered by Brock and Fries, and it has been an important tool in the molecular biology technique. This is one of the thermostable enzymes used in the PCR reaction to amplify the DNA sequences, and its special property is it can withstand up to higher temperature, up to 95 degrees Celsius. So this is one of the thermophilic bacteria. So this is from the thermophilic bacteria, and the answer is tag DNA polymerase. So moving on to the next question, DAS is the source of, which is the source of Bt toxin gene used to genetically engineer insect resistant crops.
I
Among
think your
this,
screen is got frozen.
what is the option?
Your screen is frozen.
Hello? Am I audible? Screens are visible? Whether screen is visible? Hello? B. Bacteria thuringiensis. Okay. BT toxins. Any other answer? B. You just check once. It is Bacteria thuringiensis or Bacillus thuringiensis. So we will see the detailed solution. So Bacillus thuringiensis is the correct answer, and we will see how. So it is a gram-positive spore-forming bacteria that synthesizes this toxins, BT toxin, which is parasporal crystalline, which contains Cry and CYT proteins, which are responsible for making this toxins. And this toxins has been used as a bioinsecticides against caterpillars, beetles, flies, which includes mosquitoes and black flies. So the answer is Bacillus thuringiensis, which is the source for Bt toxin gene used to genetically engineer the insect resistant crops. So how does it resist? So what happens is the leaf which has the capacity, which is genetically modified Bt crops, which has the capacity to synthesize this toxins. If suppose some caterpillar is consuming these leaves, first what happens is this toxins will be ingested by the worms, and what happens due to the changes in the pH in the gut of earthworm, the protoxin will be converted into the toxin. So it gets activated by the proteoza that are present in the gut of the earthworm. So once this toxins has been activated, so it interacts in the cell membrane and it increases the permeability. So they will form a pore on the surface of the earthworm and eventually it dies. So, so BT toxins, it acts as a bioinsecticide against various insects like caterpillars, beetles, flies, which includes mosquitoes and black flies. So Bacillus thuringiensis is the source of BT toxin, which can use to genetically engineer the insect resistant crop. Here we are modifying the crops by genetically engineering. So this is the answer. Yes, E. The option is E, not B, E. So we will move on to the next question, which is the cleaner production refers to manufacturing products with less pollution, manufacturing bioproducts, industrial production of chemicals with biodegradable waste, or all of these. Cleaner production refers to cleaner production refers to manufacturing products with less pollution, manufacturing bioproducts, industrial production of chemicals with biodegradable waste, or all of these. Yes, it is option A, because the term cleaner refers to we are cleaning or erasing the pollutant. We are not producing any chemicals, so manufacturing the product with less pollution. It is a company specific environmental protection initiatives which tends to minimize the waste and emission, right? So the answer is manufacturing the product with less pollution. So if you are manufacturing the bio product, then it comes under the biotechnology. If you are mainly focusing on manufacturing the bioproduct. So this is also industrial production of chemicals with biodegradable waste. So this also comes under the biotechnological aspect. But if we are focusing on cleaner production, it is manufacturing the product with less pollution. So the answer is A. So we will move on to the next question. So which of the following is incorrect? Option A, the environment includes both living and non-living things. Option B, biotechnology uses microbial, plant and animal cells and their product. Option C, mitochondria and chloroplast resembles the prokaryotic cells. Option D, fermentation technology is a new invention arising after the establishment of germ theory. Option E, microbial biocatalysts are generally cheap and environmental friendly. So what is the option? So which of the following statement is incorrect, which is false among the five statements? Which of the statement is incorrect? D. Okay. We will see the answer. The answer is C. Answer is D. Sorry. The answer is D. Yes, you are all correct. Answer is D. Because fermentation technology has been arised in the ancient times, whereas germ theory arises around 1500 only. So the option D is incorrect. So we will move on to the next question, which is what is the key goal of environmental biotechnology, which does not include, I will repeat, key goal of environmental biotechnology does not include which of this? Replacing plant and animal product with microbial alternative, prevention of environmental pollution, generating valuable resources for human society, cleaning up contaminated environment, designing sustainable alternative to fossil fuels. So which does not include in the key goal of environmentally biotechnology. Any answer? I will repeat it. Key goals of environmental biotechnology does not include. Any answer? Key goals of environmental biotechnology does not include. Option A. Any other answer? Option A. Okay. Any other? Okay, that is correct, Abhija. Option A, replacing plant and animal product with microbial alternatives, because this comes under the biotechnology aspect. As we have seen in the week 1 of the lecture 1, Professor has clearly stated the key goals of environmental biotechnology, which includes prevention of environmental pollution, cleaning up contaminated environment, generate valuable resource for human society, as well as producing renewable energy from the biomass. So these are all the key goals of the environmental biotechnology, and replacing plant and animal product doesn't fit into the key goal of environmental biotechnology. So we will move on to the next question, which is the modern society faces many challenges which are interconnected, but the root cause can be traced to which one of these? Mass extension, energy storage, overpopulation, food security, and disease outbreak. I will repeat it once. Modern society faces many challenges which are interrelated, but the root cause can be traced to any answer? I will repeat it once. Modern society faces many challenges which are interrelated, but the root cause can be traced to which one of this? Is it mass extension or energy storage or overpopulation or food security or disease outbreak? The answer is anyone? So the answer is option C, overpopulation. Though these are all the problems that are facing the modern society, we have seen, we have learned in the week one course that the primary problem that we are arising is rapid increase of population density. So the primary cause is population density, that is overpopulation. And rest are all like mass extinction, energy storage, food security, disease outbreak. These are all secondary crises that has been discussed in the week one lecture. So the answer is overpopulation. So the last MCQ, which is earliest study on the environmental biotechnology, were concerned mainly with the earlier studies on environmental biotechnology were mainly concerned with wastewater treatment, enhanced oil recovery, valorization of waste, bioenergy, or synthetic biology. So, which focuses on earlier studies on environmental biotechnology? Any answer? See, earliest studies on environmental biotechnology were mainly concerned with, among this, which has been focused on earlier stages of the environmental biotechnology. Option A, option C. Okay. So any other answer? So we will see the answer. It is option A. So how option A, wastewater treatment, as we have seen in the week one lecture, professor has discussed extensively on the journey of environmental biotechnology, where we could see at the earlier stage wastewater treatment, remediation of groundwater treatment, and landfill waste management were in the earlier stages of environmental biotechnology. Whereas this enhanced oil recovery, bioenergy, valorization of waste, synthetic biology that has been advent after the exploration of wastewater treatment. So the answer is wastewater treatment. So the next MCQ, which one of the following belongs to biotic component in an ecosystem? Which one of the following belongs to biotic component? Whether it is pH, dissolved oxygen, E. coli, temperature, minerals. Which one of the following belongs to biotic components? Any answer? E. coli. We will see the answer. Yes, it is E. coli. As we have discussed in the week one lecture, what are all the abiotic components and what are all the biotic components? All the living organism that directly or indirectly affects the organism or comes under the biotic components. So the E. coli is the biotic components, whereas pH, dissolved oxygen, temperature, minerals are non-biotic components. So, so this is the end of the lecture, where we have first discussed the recap of the week 2 lecture of environmental biotechnology, and second part we have seen some of the MCQs, sample MCQs, and you have answered it well. So thank you for your patiently listening to my lecture. And if you have missed out my week 1 lecture, it can be accessed. It can be accessed through the recording sheet that is there in the NPTEL portal of the environmental biotechnology. And if you have any doubts, you can post your questions in the discussion forum. And if you want to revisit these videos and if you want these PPTs, it will be uploaded in the recording sheet and you can access in the environmental NPTEL Environmental Biotechnology course in the NPTEL website. So thank you all. And if you have any doubts, do you have any doubts in this session? And you can also view this video in my YouTube channel Ajit Kumar underscore V, where you can access these videos, or if you are unable to find out it, you can always navigate to the NPTEL video where you will have a live interaction session separate tab. You can view there also. And if you want the slides, you can download it from there. So do you have any questions? Any doubts? So if you do not have any doubts, I think we can close the session. Once again, I thank everyone for listening to this session. Thank you all. Now you can leave. Thank you. Thank you, Abhija. Thank you all.